Okie dokie. So here we are in fatty acid catabolism. All right. So me forgetting my connector. Okay. So I think where we had finished up was somewhere on slide 15. But just as a quick review, because I know it seems like it's been forever for me. Um, we're talking about fatty acid catabolism, and basically how when you consume lipids, they go into the portal vein in the small intestine and get absorbed in there, and you'll see chylomicrons that come into the come on into
I think
the
it's so cool that this class lines up exactly with my nutrition class. Like they're talking about the exact same thing.
that is so helpful and so good for reinforcement. I had a advanced calculus course and a physics math course, and they were the same thing. The only thing that was different is they switched phi and rho for their abbreviations in like a certain coordinate system. And but it was great because to study for my physics math, I would take my exam for my calculus course, and it lined up. So good. But we're looking at chylomicrons and these various other small particles in here that we'll talk about a little bit. Bless you. And we'll see that apolipoproteins play a huge role. In fact, there's a chart in here that tells you what all the apolipoproteins do, but we're not going to sweat that too much. I think we had left off talking about glycerol from the triacylglycerides in there. Glycerol gets technically the fatty acids get cleaved from the glycerol, and the glycerol can then go into glycolysis. So it basically kind of goes into the DHAP route in there. So that kind of like step 4, step 5 in there. Glycerol can feed in to that because it's a 3-carbon carbohydrate, and it can make the dihydroxyacetone phosphate and get converted into glyceraldehyde 3-phosphate, and then continue on down the glycolytic pathway. Okay. Alrighty. So this brings us to this slide number 15, where we're talking about the transport or the attachment to the phospholipids. And again, we're going to see this coenzyme A play a big role here because we're going to take our fatty acids and attach them to a coenzyme A to make our fatty acyl CoAs. Okay, so we basically see on here we've got this nice little 2-step process, right? Maybe it's more of a 1-step. Yeah, okay. But again, we're going to see that our acyl CoA is going to interact with that adenosine. Lost my train of thought here. Thioltaxide, yeah, it is a 2-step process. Okay, but we're going to basically make this fatty acyl CoA compound in here, and this is a favorable interaction, very highly exergonic, so it's going to move forward. And this is how we can help transport them, because we make these very hydrophobic compounds a little more soluble, a little more hydrophilic with that addition of the coenzyme A in there. Okay, so if we look at where the oxidation takes place, and here we can talk about our carboxylic acid TCA cycle. All this is going on in the mitochondria. Same thing with our fatty acid catabolism. We need to get these fatty acids into the mitochondria so we can transport them in the bloodstream via our LDLs or IDLs, VLDLs, HDLs, chylomicrons, whatever. But we get them to the heart or the muscle or wherever they need to go. Then we need to get them into the mitochondria. Okay. So the fats get degraded into their fatty acids and glycerol in the cytoplasm, but the fatty acids have to get into the mitochondria to be oxidized and provide energy in here. If you have a very small fatty acid, like less than 12 carbons, they can freely diffuse across the mitochondrial membrane. So small ones have no trouble. They can just kind of squeeze through the holes in the mitochondria. Larger fatty acids have to be transported via the acyl carnitine transporter. Okay, so if we look at our carnitine down here, you do not need to memorize the structure, but we have this nice little carnitine molecule. Kind of looks a little bit like an amino acid in some ways, not quite, but that's our carnitine molecule. And we're going to see that we can acylate again our carnitine, right? And then we have two transporters in here. We have the carnitine acyltransferase 1, and then we have our carnitine transporter on the inner mitochondrial membrane. So basically, we attach our fatty acid to the carnitine. It goes through the transporter, and then, through the transferase and then through the transporter to get into the mitochondrial matrix. And then our acyltransferase 2, I guess you can think of as 1 is the first one it goes through, and 2 is the second one it goes through. And their mechanisms are a little bit different. The neat thing that we'll see, acyltransferase is inhibited by malonyl-CoA. All right. So malonyl-CoA, which we'll look at later on, is our first step in fatty acid synthesis. Okay. So if we are synthesizing fatty acids, we don't want to be breaking them down. So we won't bring them into the mitochondria. So our malonyl-CoA will say, if we have high levels of malonyl-CoA, that typically indicates we're synthesizing fatty acids. So we're not going to break them down. We're going to inhibit that transport in there to break them down. Okay. So again, this is a good example of compartmentalization. Our fatty acid synthesis takes place in the cytoplasm. Our fatty acid catabolism takes place in the mitochondria. Okay. So if we're making fatty acids, we don't want to break them down. If we're breaking them down, we don't want to synthesize them. No fetal cycles in here. Okay, so fatty acid oxidation occurs in the mitochondria in 3 stages here. So the first stage is creating our 2-carbon units into acetyl-CoA. So basically our acetyl groups, we're going to have an acetyl-CoA enzyme in here. We're going to use a beta oxidation process, and we're also going to generate NADH and FADH2. Okay. So we're essentially going to oxidize the beta carbon to a thioester of the fatty acyl-CoA. Right. Step 2, we're going to oxidize the acetyl-CoA into carbon dioxide through the citric acid cycle. So what we saw in chapter 16, we're going to go in there. And in step 3 is we're going to get our ATP in the respiratory chain, which we've already talked about in chapter 19. So this chapter, we're really kind of focusing on stage 1, this beta oxidation. Okay. So the neat thing is, this is a nice little repetitive sequence in here. We essentially take our nice long fatty acid, and recall that most of our fatty acids are going to be have an even numbered carbon chains in them. So we're just going to cut off 2 at a time to make our acyl group. So we can make our acyl-CoA, and that will feed directly into the TCA cycle. And that can continue on in through the respiratory chain. So we've kind of gone backwards here. The very first thing we talked about was that stage 3 with the OXPHOS, our respiratory chain. Previous chapter, we talked about stage 2, the citric acid cycle. So now we go to stage 1, the beta oxidation in here. All right, and there's all sorts of little funky things that happen here, but the primary thing is we're just going to cleave off 2 carbons at a time to make acetyl-CoA. Okay, so this pathway, let's assume we take a palmitate, palmitic acid in here. We remove an acetyl moiety each time to make that acetyl-CoA. So palmitate is a 16 carbon fatty acid, one of the most common ones. So if we do 7 passes, then we get 8 2-carbon units. So we'll make 8 acetyl-CoAs. Why is it only 7 passes? Because when we cut the last 4, well, you got cut off 2, and we've got 2 left over. So that's the question there. And the whole key is we're going to make our acyl-CoA. Okay. Hey, come on in. I am. I forgot my. Let me pause this for a second here. Let's go back to the recording and back to the sharing of the screen. And there we are. Slide 20. Yeah. Okay. All right. So let's see if it works out this way for me. Yeah, I don't know if I like this as much, but we'll see. Okay. So again, we just have this nice little pass in here where we're cleaving off 2 carbons each time to to make our acetyl-CoA that can feed into our TCA cycle. Okay, so life is decent. Okay, so how does this actually happen? All right, well, our first step is we're going to dehydrogenate our alkane to an alkene. So our alkanes, if you recall from organic chemistry, are not terribly reactive. They're not good reactive centers. About the best you can do with them is burn them, or oxidize them. So essentially that's what we're going to do. And we're going to make an alkene, which is a little more reactive. Okay. We have different isoforms of this acyl-CoA dehydrogenase, and they're really just geared to work on different sizes of our fatty acids. Okay, so if we have long chain ones, like our 12 to 18 carbons, we'll use one isoform. We've got a medium chain, we've got a short chain. Okay, we end up with a nice little trans double bond in here. So we don't normally see trans double bonds in nature for our fatty acids. We did see those with like trans hydrogen, trans fats. I don't know, you may have seen like in the 90s, basically they were very shelf stable. You didn't have to worry about them going rancid or stuff like that. So a lot of fast food restaurants would use them in the 90s. And then it was discovered that trans fats tended to build up in the arteries and greatly increased the likelihood of cardiovascular disease in people who ate a lot of trans fats. So every restaurant phased them out really fast. Can't remember if it's in these. I think maybe it's the previous generation of slides, and they talked about KFC. And it's like, no major chain restaurant uses trans fats anymore, because But we're actually going to make a trans double bond here because that makes it a little bit easier for the next step to occur. Okay, this is very similar to our succinate dehydrogenase reaction. I know we've talked about a couple different dehydrogenases. The mechanism is very, very similar for all of them. Let me slide this out of my way here, hopefully. Okay. And of course, we're going to take these bound electrons and transfer them to our FAD so that we get our nice little generator. Come on, there we go. Okay. So here's our FAD cofactor. Again, you can see these lovely little ring structures up here. The ring structures are great for electron transport in there. And again, we end up with the semiquinone, and then we can fully reduce it with a second electron to get our quinone form. Okay, so we go from our, in this case, the isoalloxene ring, but we're basically making our semiquinone to our fully reduced FADH2. Okay, so it's a nice little two-step process in there, very similar to the ubiquinone that transfers electrons from complexes 1 and 2 to complex 3. Okay, so we've got our first steps in here, and in our second step, we can see that we're going to use NAD as well. And when we're going to have our NAD dehydrogenase to our respiratory chain, again, both our FAD and our NADH end up going to our respiratory chain there, providing electrons that way. Oh, all right, wherever I put this, it's always the wrong spot. Let's try up here. Okay, yeah. All right. There we go. Okay. So the second step, after we have dehydrogenated it and made that double bond, we're now going to hydrate it. Okay, so we take away 2 hydrogens and we add a hydrogen and a hydroxyl group. We're going to add water to it. Okay, so the water adds across that double bond, giving us our nice little alcohol on the beta carbon. Okay, so that's going to be basically our second carbon in there. This is going to be very similar to the fumarase reaction in the citric acid cycle. It's got that same stereospecificity, same type of mechanism in there, and we get that alcohol. Okay. The neat thing is, by adding that alcohol to the beta carbon, we've now activated that beta carbon, so we can make it easier to cleave. So we can dehydrogenate the alcohol. Here we have this a second dehydrogenase in here using our NAD cofactor. We get that nice little step in there. All right, so now we're down to here, our NADH moving up this way. All right, so now we have made our ketoacyl CoA, and now we're going to bring in a second coenzyme A to attack this carbon here, and we will cleave off the acetyl group that we worked so hard to get those oxygens into to make our acetyl CoA. Okay. So that's our step 4. So our 1, 2, 3, 4. We have this acetyltransferase, sometimes called a thiolase, because we're using that thioester bond from the coenzyme A that will attack that carbonyl carbon in there. And we pick up the fatty acid chain. So we kind of cleave off the end, acetyl-CoA, and add a new CoA in there. So we now have an acetyl-CoA that we released, and a new fatty acyl-CoA that is just 2 carbons shorter. And then we repeat this step over and over again until we've turned all the carbons into acetyl-CoA. Okay. All right. So this is a lot of reactions, right? We got basically 4 reactions that are going on here: 2 dehydrogenases, our transferase, and our hydroxylase. What is it? Hold on. Hydratase is what they call it. Okay. Okay. All right. The cool thing is it's a single enzyme that does a single protein in here. So basically it's this big trifunctional protein with these multiple subunits in here that will have all sorts of reaction. The neat thing about it being a single trifunctional enzyme, easy to regulate, and you have all these processes right beside each other. So it can be this nice little assembly line that's going down the chain there too. And then as we shorten our fatty acid group, we can pass it on to our different isoforms. So again, I think we had discussed substrate channeling. If all of our different enzymes are held really closely together, I think we saw this in Biochem 1 with scaffolding proteins that will hold them together. We'll have all these nice and close together. And our trifunctional protein is associated with the inner mitochondrial membrane. So it's, I believe, anchored in there. And then shorter chains can be processed by the more soluble ones in the matrix. Okay. All right. Yeah. Basically, what we're looking at here is this is a, common procedure in here. Okay, so you've got glyoxysomes in your plants, you've got your gram-negative bacteria, gram-positive bacteria that have, and of course our long chain specific section here that's bound to the inner membrane. All of these are held closely together. This is a common reaction scheme across organisms. So very important, very old. It works. Okay. So let's talk about what we've actually done. Besides our mechanism here, we are going to take, say, palmitic acid, our 16 carbon fatty acid, and just cleave it into 8 separate acetyl-CoAs. Okay. So for palmitic acid, we repeat this step a bunch of times. We get 8 molecules in there. We end up making 7 total FADH2s and 7 total NADHs. And then our acetyl-CoA can go into the citric acid cycle. And you recall in the citric acid cycle, you get a GTP, an NADH, and an FADH2. And all these electrons from our FADH2 and NADH enter the electron transport chain. And our GTP is functionally equivalent to an ATP. Okay, so this is why if you take 1 palmitic acid, okay, in essence, we can get about 108 ATPs out of it. Okay, so we're assuming 1.5 ATPs for each molecule of FADH and 2.5 for each NADH. Right, and our GTP we're assuming is equivalent as well there too. Okay, all right. So I'll kind of leave it to you guys to kind of look at those calculations in there. I think I might have a question where I say, hey, justify why this is accurate or not, in there, but we're not going to sweat it too much. But the idea is that our fatty acids can produce a lot of energy. Okay, so if we, burn 1 palmitic acid, we get 108 ATPs. So lipids are great sources of energy. Storage, we kind of mentioned this before. They're easy to store in the body. You don't have a lot of excess water in there. And then when you do burn them, you get a lot of energy from them. So evolutionarily, we've developed a taste for lipids. It's like, why does bacon taste so good? Because of all the fat, because our bodies crave fat. Because if you go back to, ancient times when it was kind of like hunter-gatherer society, if you killed a buffalo, killed a dinosaur, and everybody eats. And if you go through lean times, you have to store that energy for when you do eat for later. And if we look at, say I was at the zoo, so we looked at the bears and they all looked healthy and they're well fed. But I keep thinking of the bears that, getting ready for hibernation. And they have like the what's it, Alaska has the fat bear contest where they show you all the bears that are putting on their hibernation weight so they can get through the hibernation too. Well, they have to eat a lot, so they have all those fat stores. So while they're hibernating for several months, they can burn that fat. And then you have skinny bears that come out of their dens in the spring and start doing their bear things, looking for the bare necessity. Okay, no, stop. Sorry, my mind works in weird ways. So evolutionarily, we have a taste for lipids because it's a good source of long-term energy. Okay. All right. This mechanism is actually very similar to a lot of other mechanisms that we see. Our beta oxidation, similar to our citric acid cycle, similar even to our oxidation of some amino acids like isoleucine. So we can see that we're producing these FADH2s and NADHs that can feed into the respiratory chain to produce energy. Okay?
Did you say that it results in 108 ATP?
Yes.
What slide is that
on? This is, what slide is this? This is slide 32.
Oh, okay.
Yeah, yep. Okay. So, again, just to kind of summarize this pathway, the similarity, this is a pathway that works, that produces energy. So we see it mirrored in a lot of other processes as well too. All right, so palmitic acid is a nice, simple, saturated 16-carbon fatty acid. What happens if we have an unsaturated fatty acid, if we have a double bond in there, okay? Because those are naturally occurring, and they're typically not a good substrate for enol-CoA hydratase in here. So we see a couple extra enzymes that get involved in here. Keep in mind that our naturally occurring unsaturated fatty acids typically have cis double bonds. They introduce that kink into the fatty acid so that it has a bend in there too. And our trifunctional enzyme here is actually making a trans double bond. So what we need to do is convert that cis double bond into a trans double bond. Okay, so we have an isomerase to do that, and then our reductase will reduce any cis double bonds that are not at carbon 3. Okay, so if we have them at like the wrong position in there, and we'll just get rid of them and saturate the fatty acid and then proceed as normal. If we do have a double bond that's at the right length in there, then we'll convert those to trans double bonds. Okay, so our monounsaturated fatty acids require the isomerase. Okay, and polyunsaturated fatty acids will take both, because typically we're going to see every 3 carbons is where we have a double bond in our polyunsaturated fatty acids. Okay. All right, so if we look at something like oleic acid here, you can see we have a single double bond here at carbon 9. All right, so as we cleave off, we go through the first few rounds, and then we get here. So we have got our acyl CoA enzyme. So we've got our first carbon. Come back here. Second carbon, and our third carbon is where our double bond is. All right. We will see this isomerase come in here and convert this from a cis double bond to a trans double bond, and then we can proceed as normal. Okay. If we look at what we have here, I want to say this is linoleic acid, linoleic acid up here. Yep. All right. Stick in the corner there. We proceed as normal through here, and then we get to carbon 9, which now becomes 1, 2, 3, our third one. We can convert that to trans as normal. That's all well and good. We cleave off 2, we cleave off 2, and then we've got this weird little funky thing here. So now what we need to do is get rid of this, not only get rid of the cis double bond, but it's at the wrong position for an isomerase to work on it. So we're just going to saturate it and go across it, add hydrogens across it to get it in there. Okay, and then we'll have our isomerase for our last one. Okay, all right, all right. So all well and good. All right. So basically, more complex fatty acids require the addition of a few more enzymes. Okay. Every time. Okay. And then this is just that in detail. Okay. So that works for all of our even numbered fatty acids. Okay. Most dietary fatty acids are even numbered. Palmitic acid, the most common one out there. But occasionally we run across some odd numbered ones. So, we're cleaving off acetyl-CoA, 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 acetyl-CoA. And then we get down to that last little section where we have 3 left over. And now we have a 3-carbon compound. We make propionyl-CoA. Okay. So, we have propionyl-CoA in there. We can also see this in various animals that like chew their cud, like the water buffalo that we saw at the zoo, or deer, or cows, or whatever in there. Okay, our propionyl-CoA is going to be oxidized in a slightly different way. All right, so what we're going to see here is we're going to take our propionyl-CoA, and we have this lovely little propionyl-CoA carboxylase, which will add carbon dioxide to it. This is coming from bicarbonate, and we'll make methylmalonyl-CoA. All right, and we actually, our stereospecificity, we end up with dimethylmalonyl-CoA, so we're going to convert that into L-methylmalonyl-CoA, and then we mutate that around. We kind of flip our coenzyme A to be attached to the different carbon here. Yep. We're going to hook this over to the end to make our succinyl-CoA. All right. So propionyl-CoA, it doesn't feed directly into the TCA cycle the same way as the others do. This last little section of odd numbered ones is going to ultimately be converted into succinyl-CoA. Okay. And you can see here, once again, our propionyl-CoA carboxylase, we're adding a carbon to this. So we're going from a 3-carbon group to making it a 4-carbon group. Right? You think if you make 4 carbons, you just cut that in half and get 2 acetyl-CoAs. Nope, going a different way. But look again, we have a cofactor of biotin in here. Remember, our biotin has that long arm, so that is very useful for moving. It's a very common cofactor for methyl transfer in there. So as we move that methyl group that's coming, basically our carbon dioxide that's coming from our bicarbonate, we can put that in the right position to interact to make our methylmalonyl-CoA. All right, we end up mutating that to L-methylmalonyl-CoA. Do I see a big difference in there? Yeah, okay. All right, slide that around there. And then for our last step, we're essentially going to shift this over to the last carbon and basically move this carbon up here. So again, we're moving a single carbon unit, just we have a CoA attached to it to make our succinyl-CoA. Okay, and our succinyl-CoA can then further go into our various metabolic pathways. Hey, it was a long day yesterday. I'm tired. Okay, so again, besides our biotin, coenzyme B12 in here, our vitamin B12. Whenever we have one of these transferases or these isomerases, we'll often see this being necessary. So B vitamins tend to play an important role for as cofactors in these reactions. Okay, so that's why you'll often see, especially in like neural tube development for a growing fetus. This is why probably one of the most important health regimens for pregnant women is prenatal vitamins. And you'll see now pretty much, I think just about any woman who goes to OBGYN for pregnancy, it's like, here's your bottle of vitamins. They'll just hand you a bottle of vitamins in there because you need your B vitamins. Lots of leafy greens, you can get them that way too, but supplement with this just for everybody's diet is somewhat different, so that you can get that neural tube, proper neural tube, neural crest formation, and good brain development to help avoid any abnormalities in the unborn child. All right. So what does vitamin B12 or coenzyme B12, depending on how you want to look at it, look like? This is our cobalt containing compound. So this is one of our neat little oddities in here. We've got cobalt as a cofactor in here inside this corn ring system. You might look at this corn ring system and say, hey, that looks a lot like the porphyrin ring system for hemoglobin and all that. It has some similarities. We do have a nice big, essentially flat and heavily conjugated ring system in there that can help coordinate this cobalt. And then we have this deoxyadenosine above the cobalt and a dimethylbenzimidinol ribonucleotide below it. All right. And again, this is just very useful. You can see based on the structure, they obviously are going to be passing. This is a good electron sink. All right. So we got a good electron sink in here. All right. So if we have a nice little electron, we can have an electron withdrawing, electron donating. I won't emphasize this mechanism very much, but this is a good thing to look at to kind of understand how this works if you want to go into great detail. Right? We're not going into great detail at this level, but vitamin B12 is important for functional group exchange because of that electron withdrawing property. You can see here that we can actually make this substrate radical. So we have some radical chemistry. For fun, one of my professors, when I was a student, was talking about in the late 60s, it was either 68 or 69, he went to Chicago. I think it was 69 because it was the year after the Democratic Convention where they had all the riots in Chicago. He went there for the Association of Radical Chemists convention or whatever. And, word radical, in chemistry means you got a single electron, right? But this is right after all this political unrest. So, the police and the FBI were investigating them because they're a radical chemist. Oh, they're going to blow stuff up. But they're like, no, we're just talking about single electrons. So, our cobalt containing compound, our vitamin B12, can facilitate that single electron reaction. Okay, plants. We're mainly focused on mitochondrial dehydrogenase in the electron transport chain. Plants do something similar, and that's all I want to say about that because we're not going to talk about plants at all. All right, so we can basically skip these slides in here. Well, the next two slides. All right. So if we jump ahead, all right, once we make our acetyl-CoA, just like we saw in chapter 16, this can go into the TCA cycle, and it reacts with oxaloacetate, our OAA. Okay, if we run low on oxaloacetate, then the acetyl-CoA will instead get converted into ketone bodies. Okay, and there are three of these that are most commonly seen are acetone, acetoacetate, and beta hydroxybutyrate that are depicted here. So we basically, this conversion into ketone bodies allows coenzyme A to be freed up so that it can continue beta oxidation, right? So if you still need energy, but you don't have enough OAA, then you make ketone bodies and you can still metabolize fat, all right? These three can leave the liver and they can provide energy to the brain and heart and muscle, all right? All right, so do I want to talk about this? Yeah, we'll get there. Okay. So how do we form these? All right. So if we look at our acetyl-CoA that we have here, we can actually take two acetyl-CoAs and we can kind of combine the acyl group from one with the acyl group on the other to make this acetoacetyl-CoA. All right. So there we have our acetyl-CoA. All right. If we have that interact with acetyl-CoA and catalyzed by HMG-CoA synthase, which we will see much more of later when we talk about fatty acid synthesis, we can make a beta hydroxy beta methyl glutarol-CoA or HMG-CoA. Okay. All right. So essentially, we are kind of reversing the last step in the beta oxidation. We join together two acetate groups. Instead of making all these 2-carbon units, we're going to take the last two we made and put them back together. And sometimes we can put a third one in there. But the idea is we can make these, 3-carbon, 4-carbon, or variant 4-carbon.
So
groups.
Reversing the original process?
Yes, so that beta oxidation, you just cleave off 2 carbons at a time. Here, we're going to take 2 acetyl-CoA groups and bring them together to make a 4-carbon unit and free up one of those coenzyme A's. Okay. All right. Okay. But basically, once we make these CoA compounds here, we still need to get rid, free up that CoA. So from our HMG-CoA, we have this HMG-CoA lyase that will cleave off the acetyl-CoA, and then we can have either acetoacetate, which can be further broken down to acetone, or it can be interconverted to beta hydroxybutyrate. Okay. All right. So, yeah, okay, we'll get there in a minute there. All right. So we make our HMG-CoA, which we're going to see in fatty acid synthesis. So that sort of makes sense that if we're kind of reversing that last step of the fatty acid oxidation, that's going to be our first step in synthesis. But rather than synthesizing lipids, we're going to remove acetyl-CoA and then produce these ketone bodies. So the ketone bodies are made in the liver, okay? And then we can transport our acetoacetate and beta hydroxybutyrate through the bloodstream, and they can be used as energy for the heart, muscle, kidneys. I always forget the kidneys, and the brain. Okay. Our coenzyme A, goes back in whatever fatty acids we do have available, whatever oxaloacetate is available. We can still keep, producing, running the citric acid cycle in there. If we make acetone, that actually gets exhaled as a gas. Okay. Alrighty. If we use these as fuel, we do here. Yeah, there they are. Okay. All right. Wow. This chapter seems short. One of the things you'll note, anybody familiar with the Atkins diet or the low carb / no carb diet? Yes. Yeah. All right. Biochemically, this works. This is a great thing. You eliminate carbs, you're going to eliminate, a lot of oxaloacetate in there, and it's going to stimulate your body to burn. You're also going to not have a lot of glycogen as well. So, but you're going to kind of put your body into a starved state, so it starts burning lipids, right? One of the things that happens, some people would often overdo it and completely cut out all carbs right away, and then they'd essentially go into like a diabetic shock, essentially like that, because of the rapid turn to ketone bodies. One of the things that you'll notice, people with low blood sugar who have diabetes, sometimes their breath will smell like acetone, right? And that's because they're actually making all these ketone bodies, including acetone, which is being exhaled. So people who go on a very strict form of the Atkins diet will often be producing so many ketone bodies, they'll be exhaling acetone as well too. So they'll have that same type of odor to their breath. It's my opinion that a very low carb diet is probably a little bit safer than a no carb diet, because you can certainly survive on ketone bodies, but then you start the risk of ketoacidosis of the bloodstream and changing the pH, and coma, death, all that sorts of good stuff in there as well too.
I saw this girl on TikTok, and she didn't eat anything but ground beef soup. She called it ground beef soup, but it was nothing but ground beef and the fat that she cooked it in.
Yeah.
And she just ate it for a week. That was so gross.
Yeah.
We learned in nutrition class too that the red blood cells can't use anything but glucose.
Exactly. Yep.
for
Exactly.
energy because they don't have mitochondria. So if you do no carb, you essentially would die of hypoxia.
Yep. So you can, So even if you're eating, so it's balance. I always laugh. It's like, I eat a high carb, high protein, high fat diet. Wait a minute. That's not gonna work.
And then if
balanced.
French fry, you're like,
Yep. But I know there's still people who go to fast food restaurants and order like a burger with no bun and stuff like that. But that was when the Atkins diet first came out. I remember that was a big thing, and all the fast food restaurants were advertising, you can get it without bread, or like a Subway sub without the bread. And I'm like, well, it's called a charcuterie board, or
Well, now the big fat diet is the keto diet, which is essentially really unhealthy because it's high fat, so you're just clogging arteries.
Yeah.
So you may lose weight.
so nasty. Like
It's
their food looks so bad.
yeah.
Like instead of using flour for fried chicken, they fry their chicken with pork rinds. That sounds even
Oh
worse.
man. Well, see, I can't complain too much. So when we have duck for Thanksgiving dinner, and
That's like
the duck fat that comes off of there too, I collect the duck fat and then we fry it. Like it's so greasy, but it's so good because you fry potatoes and duck fat. I'm like,
That's
oh
why
my
I
lord.
don't like fancy restaurants because I can't ever eat. They always want to put duck fat in there.
Yeah, yeah, yeah.
Like I can't even eat green beans here.
Yeah, but and there's all sorts of shortcuts too, because one of my little time wasters and take a break, a little reward for grading and stuff, something like that is little those little downloadable games, something like that. But they show the ads, and one of the ads that's showing up now too is like the magic fat burner, as on Shark whatever or something like that, and they just it's like no, it doesn't work that way. You can't just it's hard, but everything is diet and exercise. That is probably the healthiest way to control one's weight. And it's hard. It's challenging. I know for me, it's like, oh, let's cut out Mountain Dew a little bit. And it's like, oh, it's like that's hard. I'm a hardcore addict right here, straight into the veins.
It used to be me.
Yeah. I mean, when I was a freshman, I had scholarship money, so I had like a 2-liter Mountain Dew and like a big bag of Doritos pretty much every day.
Yes.
Yeah.
That's really the best combination.
Yeah. But now that I'm older, my metabolism's slowing down. It's like, oh, I need to cut this out. It's challenging because where am I going to get my caffeine? I never learned to drink coffee. All right. Anyway, so we're at the end of chapter 17, which is good. Here we're really looking at our oxidation of fatty acids. Okay, so we're pretty much going to try and cleave everything off 2 carbon units and just feed that into the TCA cycle. All right. If we have, like there's basically a trifunctional enzyme with 4 steps to cleave those off. So that's a good thing to know. Those kind of 4 steps in there and all that. If you have, an unsaturated fatty acid, then you have to add in some extra enzymes, 1 or 2 extra enzymes. If you have an odd numbered fatty acid, then you end up with a propionyl-CoA, which gets converted into succinyl-CoA. All right. And of course, if we run low on oxaloacetate, then we can make our ketone bodies. Okay. And that's it for this chapter. It's a kind of short chapter. I don't think this chapter is that bad, and it's fairly intuitive. So I'm looking towards the upcoming exam, which will probably be the last week of March, probably in about 2 weeks or so. It's usually not too bad. The second exam is usually probably the better
You always
exam.
say that.
Yeah. Well, the second exam in here is usually probably one of the better exams. It's either the second or the fourth. Those are probably the 2 that students do the best on. Okay. And
But you have to basically memorize the Krebs cycle for this
yes,
test, right?
yes. Yeah. And that's a pain. That's true. That is a pain. But once you do memorize the Krebs cycle, when we're actually going to practice that, probably not this week, but probably next week. I don't know, either next Wednesday or maybe next Monday. I'll have to look and see where we are. But we'll do a little practice on that as well too. And then I have an exercise for you guys for probably in class where we will look
I would
at
like to see
making us
write
some
the
questions.
entire thing on the whiteboard.
Exactly. Yeah. Actually, you know what I've done in the past? I've had students come, you draw this part, you draw this part, you draw. Yeah. So yeah, we'll do something like that. Yeah. It's actually a good habit to get into. And with only 3 people, it's easy to get here to the board and draw. And next time I forget my thing again. Okay. But what do I do here? Stop recording.